her time in the Weiss lab perseverance. Angela has had a very rough road in terms of experiments in the Weiss lab, but she's triumphant in the end. So for all of you youngsters out there, never give up. And if it's hard to get discouraged, talk to Angela. Angela started in 2008, and her interest almost immediately was in studying epigenetics and its role in symbiosis in coronal bioplastic symbiosis. And this arose out of taking a course from Michael Freitag, who is on the computer from Germany. He's on Skype. Hello, Michael. So she started off on all these, she had these great ideas. We all thought it was wonderful. And she marched along, and it was a complete and utter catastrophe. And so after about, I don't know, a year, I pulled the plug and said, we're done. So then she went back to the drawing board, and she did some really nice phylogenetics work that she got skipped on. In her lab, she came out with this beautiful paper that was exactly her data, that she was about 75% fit. Okay, so then she started down the path of PhD, and interested in complement. She can tell you all about that. And she was on the path of studying one of the central genes that she described. And I went and visited my colleague in Stanford, John Tringle, who was also working on a PhD. And the first student, Liz, doing her thesis work on C3 complement, and was further along than Angela. So that was strike free. But it's all good, because she has a beautiful thesis, and she's going to tell you all about it. She's also, along the way, became very, very interested in pedagogy and teaching. And many of us know that because we saw her interview here for a job and did extraordinarily well there, and is actually starting a job last week at Western Oregon University teaching in the biology department there. So there is no rest for her. She is starting almost probably tomorrow on her duties up at Western. So we will miss you so much, Angela. She's been wonderful for me in the lab. She is the lab mom. Sheila, get to work. Get to work cut out for you. But you're a fantastic person, and I'm very excited for you. Today she's going to talk about the role of innate immunity in Cnidarian dinoflagellate symbiosis. Angela.
Thank you, Virginia, and thank you all for coming. I know 9 a.m. is a little bit early sometimes on summer mornings. And so as Virginia said, I'm going to present to you my thesis research, the role of innate immunity in Cnidarian dinoflagellate symbiosis. And so just a little roadmap for the presentation today. I'm going to start with a pretty lengthy introduction to get you all up to speed on this topic, talking about Cnidarian dinoflagellate symbiosis, the role of immunity in symbiosis, and then also the complement system, which is my focus for today. And then I'm going to share 2 of my chapters with you, the role of complement in symbiosis, and then complement and immune challenge, and then I'll end with some general conclusions. And so I'd like to start our discussion of symbiosis by just talking kind of broadly about the symbiote interaction. And so symbiosis refers to basically any sort of partnership between 2 or more individuals. And this can take on a variety of natures. It can be a parasitic relationship in which one organism or one partner benefits at the expense of the other. It can be a commensal relationship in which one partner benefits and the other isn't really affected. And then lastly, it can be a mutualistic relationship in which both partners benefit and derive some sort of novel capabilities from the relationship. And so I'm going to focus in on a particular mutualistic relationship today, that between cnidarians and the symbiotic dinoflagellates. And so again, this is a mutualistic relationship between cnidarians, such as corals and sea anemones, and dinoflagellates of the genus Symbiotic. And so to give you a little bit about the anatomy of this relationship, and so this image here represents a portion of a coral head. It's made up of genetically identical units called polyps. And so if we look at a polyp, you'll notice it looks very much like the body of a sea anemone. And we're going to be seeing a lot of that today. And then if we look at this blown-up image of one tentacle, we begin to see the tissue layers. So cnidarians are diploblastic organisms. So they have an outer epidermis, which is this clear layer here, and then an inner gastrodermis that surrounds the gastrovascular cavity. And it's in this gastrodermal tissue that the symbionts reside. And so you can see all of these little gold spheres in here are the dinoflagellates. And this relationship is based on nutritional exchange. And so the symbionts are intracellular within the host cell, and they are photosynthetic. And so they are transferring photosynthate or sugars to the host cell, and then in return they receive a stable intracellular environment to live in, and also inorganic nutrients such as nitrogen and phosphorus. And so why study this relationship? First of all, it's of great ecological significance. And so this symbiosis really forms the basis for the high productivity and diversity on the coral reef ecosystem. And so you might imagine that the breakdown of symbiosis can be highly detrimental. And so the breakdown is commonly referred to as coral bleaching. It results from a variety of environmental stressors such as rising sea surface temperatures, salinity, pollution, disease. And so it's called coral bleaching because the coral takes on this kind of white or bleached appearance in contrast to the healthy coral, which has the pigmentation from the symbionts. So coral bleaching is loss of symbionts and their pigmentation. And so coral bleaching can really kind of decimate the entire reef ecosystem, as shown in this image here. And so understanding the kind of molecular and cellular underpinnings of this relationship and what occurs during breakdown is really important for understanding and mitigating bleaching. In addition, kind of linked to this is the fact that coral disease is increasing in both its frequency and prevalence. And so there's been a lot of work done over the last decade or so on determining kind of etiological agents of this disease, but at the kind of molecular and cellular level, again, it's very poorly understood how corals respond to challenge with pathogenic microbes and the cellular defenses they have. And so understanding both of these is really important for understanding coral health. Studying Nigerian dinoflagellate symbiosis is also of evolutionary significance. And so Nigerians are basal metazoa, so this is the eukaryotic kind of bush of life, and so metazoa is right here in this red circle. And if we look kind of closer at this, you can see that Nigerians are part of this basal metazoa group that is kind of sister to the Bilateria. And so they have a surprising amount of genomic complexity compared to a lot of other invertebrate groups, particularly that of protostomes. And so they represent a really good model for studying kind of ancestral signaling pathways, both their evolution and their function. In addition, they're really, it's important to study in the context of host-microbe interactions. And so the symbiotic dinoflagellates, pictured here, are kind of a sister group to the apigenlexin parasites. So these are parasites of relevance to human health, such as Plasmodium falciparum, that's the causative agent of malaria, Toxoplasma gondii. And so understanding how dinoflagellates are interacting with their Nigerian host can also provide clues to how apigenlexin parasites interact with their definitive and intermediate hosts. While it is important to study coral bleaching and what's going on during the breakdown of symbiosis, it's also important to understand what's occurring at kind of the onset of symbiosis, what forms of stable symbiosis. And so, for most Nigerians, symbionts are acquired anew from the environment, and this occurs during larval stages. They're taken up through the mouth, and then phagocytosed by gastrodermal cells. And so you can see the phagocytosis process going on in this image here. And so this is a symbiont. You can see a gastrodermal cell kind of making initial contact with it, and then bringing it into the cells. And this is also a really highly specific relationship. And so Symbiodinium is an incredibly diverse genus, more on the level of an order than a genus. And hosts, therefore, will generally only associate with one or a few strains. And the real depth of this specificity is just beginning to be understood. So in my discussion of Nigerian dinoflagellate symbiosis today, I'm going to be talking a lot about immunity. And so I just kind of wanted to take a moment and define some terms within immunity for the non-immunologists in the crowd. And so when we talk about immunity, we're talking about the capability of an organism to defend itself against some sort of foreign invader. And the vertebrate immune system is broken down into two major arms. So there is the innate immune system, which is really fast-acting and is a relatively non-specific response in that you have kind of general host pattern recognition receptors recognizing general microbial signatures. And then in contrast is the adaptive or shown over the last decade that immunity can help mediate not only interactions with negative microbes, but also positive associations. So we can think about it kind of along the whole symbiosis spectrum. So this will be important for my talk. And so there's been a lot of focus on innate immunity in the white lab. And so all of these, nitric oxide production, TGF-beta, apoptosis, lichen-glycan interactions, these all represent innate immune pathways or molecules that have previously been shown to play a role in Neigerian Dinoflagellus symbiosis. And so I am going to focus on a different immune pathway, the complement system. And so I'd like to take some time now to tell you about this pathway and what's known about it in Neigerians. And so I'm going to warn you that this is a very complicated pathway, and even what I'm showing you today is kind of a simplified version. And so I'd like to start at the end of the pathway, and this is going to be for vertebrates specifically. And that the end goal here is really formation of a structure called the membrane attack complex, which leads to microbe death. And so now we're going to kind of go back to the beginning and see how we get to that endpoint. And so the complement system has three distinct mechanisms of activation. And so these are the classical pathway, the lectin pathway, and the alternative pathway. And so these all use different molecules to kind of initially recognize and detect microbes and converge at the cleavage of the central molecule in the pathway, C3. And so if we take a look at how we get there, so for the classical pathway, what we have is this protein complex of C1 recognizing antigen-antibody complexes that are on the surface of a microbe cell. And so binding of this portion C1q to these antibodies will activate two serine proteases, C1r and then subsequently C1s. These cleave complement components C2 and C4, and then fragments of these cleavage events will come together to form what's known as the C3 convertase. And this is a really critical step in the complement system because it's this C3 convertase that cleaves the central molecule in the pathway C3 into fragments C3a and C3b. So C3a kind of diffuses away, activates other components of the inflammatory response, whereas C3b will bind to the surface of the microbe, kind of coating it in a process called opsonization, and then C3b can be recognized by phagocytic receptors, resulting in phagocytosis of the microbe. For the lectin pathway, we're going to see a lot of similarities. And so you'll notice that the kind of initiation molecules are very similar in structure to that of the classical pathway. So here we have mannose-binding lectins and phycolins binding to sugars present on the surface of the microbe. And similarly, they have serine proteases associated with them. These are called MASCs, or mannose-binding lectin-associated serine proteases. And these, when activated, will also cleave complement components C2 and C4, therefore leading to the formation of the C3 convertase and then downstream C3 cleavage, just as we discussed for the classical pathway. And then lastly, the alternative pathway is a little bit different. So this is kind of a spontaneous activation mechanism in which C3 undergoes kind of spontaneous hydrolysis. It's kind of happening constantly in the cell, and then this will kind of end up eventually leading also to cleavage of C3. And here, we're going to have involvement of a couple of new molecules. So C3b will bind to the surface of the cell, and another serine protease called factor B is going to bind to that. This binding initiates cleavage of factor B by another serine protease, factor D, and then we have the formation of a second C3 convertase. So this C3 convertase, although made up of kind of different subunits, has the exact same function as this convertase that we saw for the classical and lectin pathways. And so this will also initiate further cleavage of C3 and then opsonization of the microbe. And so downstream of this C3 pathway, or the C3 cleavage, all three of these pathways are now united. And I'm going to kind of overly simplify these steps and say that we have activation of the downstream complement protein C5 through C9, and these all come together then to form this membrane attack complex that creates pores in the surface of the microbe, leading to death. And so, again, this is all for vertebrates, and so you might wonder, what does the invertebrate complement system contain? And so, it's a much more kind of simplified version, only some of the key molecules are present. And so I want to also kind of illustrate what's present and what's absent. And so you might notice that this whole formation of the membrane And then C32, although it was constant across tissue layers, showed decreased expression in symbiotic anemones. So again, this is kind of more evidence to suggest that C3 may play some role in symbiosis. So again, what's missing here is any information about factor B and MAPS in Nigerians. And so this is really what I wanted to focus my thesis work on. And so for my thesis chapter, for chapter 1, which I'm not going to be sharing with you, this is a recently published paper about tier domain containing proteins, which is another domain that's found in a lot of innate immune molecules, and kind of characterizing them and looking at their evolution in the amphizoans. So I'd be happy to talk to anyone about that at a later date. And then what I really want to focus on today is the complement system. And so for all of my functional work, I'm going to be using Aptasia, and I'll tell you a little bit about Aptasia in a moment. And then for chapter 2, what I want to do is look at beneficial interactions with microbes. And so for this, we'll be exploring the role of factor B and MAPS in Nigerian dinoflagellate symbiosis. And then kind of in contrast, on the flip side, I also, in chapter 3, wanted to look at the role that factor B and MAPS play upon immune challenge. And so we'll look at immune stimulants and also live bacteria. So before we jump into chapter 2, I just wanted to take a few minutes to talk about Aptasia. And so we use Aptasia in the lab as a model for studying Nigerian dinoflagellate symbiosis. And one reason that this is a really attractive model is that we can maintain Aptasia in both the symbiotic and ecosymbiotic states. And so you can see down here are symbiotic Aptasia that you can see the pigmentation from the dinoflagellates, and here the ethos, which are kind of white and transparent. And so having these allows us to make comparisons between symbiotic states, and also we can kind of reinfect these ecosymbiotic animals with dinoflagellates and study the dynamics of the onset of symbiosis. And then additionally, there's been, in the last couple of years, a lot of molecular resources developed for Aptasia. And so this includes a complete transcriptome, and there's also genome sequencing underway, and it's kind of currently in the analysis phase. Okay, so moving on to chapter 2. So again, this is all about the role of complement in Nigerian dinoflagellate symbiosis. And so I have several objectives for this chapter. So first, in order to do kind of any work on these complement molecules, I needed to characterize them in Aptasia. Second, I wanted to perform phylogenetic analysis on these new sequences that I acquired and other sequences from invertebrates to kind of get a sense of how they were all related. And then lastly, the functional component of this chapter is determining the expression profiles of factor B and MAPS genes as a function of three things: symbiotic state, the onset of symbiosis, and also tissue-specific expression. And so I'm going to start by telling you about what I characterize for complement sequences in Aptasia. And so what I found was that Aptasia has 3 factor B sequences. These are pretty similar. They're a little bit different in length, largely due to variation in kind of how many complement control motifs they have at the N-terminal part of the protein. And then there was also 1 MAPS sequence. And so I wanted to put these up here to kind of highlight which ones I'm going to be talking about today. And so I'm only going to be talking about 3, which include APDF1, APDF2B, and then lastly APMAPS. So I found kind of the other factor B sequence after I'd done a lot of this functional work. And so that's why I will not be I'm going to talk about kind of some general methods here for both of these experiments, and they're going to kind of come up again and again throughout the talk. And I also, before I forget, want to acknowledge Sheila Kitchen, sitting right up here. She was a big part of kind of the development of all these methods and the conduction of these experiments, and so this wouldn't have been possible without her. Okay, so for the symbiotic state comparison, what I did here was sampled steady state symbiotic and ecosymbiotic animals. And then for the onset of symbiosis, ecosymbiotic aptasia were inoculated with symbiotium and then sampled at selected time points. And so for this, I have kind of two different treatments. The first one, which is kind of what I just previously described here, is the symbiont treatment, where symbionts were added to a final concentration of about 10 to the 5th cells per mil. And then as controls, we also included no symbiont treatments. And so the reason for this is that we put these animals in the light during the infection process. And so we have to have these no symbiont controls in order to kind of account for the effect that light alone might have on complement gene expression. And so downstream of this, the analysis pipeline is pretty much the same for both experiments. So I extracted RNA and did cDNA synthesis, and then performed relative quantitative qPCR. And so these was measuring levels of transcript of APDF1, APDF2B, and APMAPs, and we normalized to reference genes L10 and L12, to ribosomal proteins, and then also poly-A binding protein. And then in the end, our analysis, the metric that we used in the end for data analysis was relative quantity. And so I'm going to share with you first the results of the symbiotic state comparison. And so on this graph, we have our genes of interest, and then relative quantities on the log 2 scale. And then the striped bars are going to be ecosymbiotic animals, and the solid are symbiotic. And so the only significant difference that I saw in this experiment was for APDF2B. And so you can see that it is significantly lower in the symbiotic and ecosymbiotic state. And so this was an interesting result and kind of the first clue that APDF2B might be involved in some way in symbiosis. And so for APDF1 and APMAPs, there was no significant difference observed, although you can see that the same trend that APDF2B showed can be seen for MAPs, but APDF1 doesn't really show any trend. And so you can notice that there's a lot of individual variation here, particularly in the symbiotic animals. And so this may be kind of masking a true difference, and this is kind of a common problem in our system, that we have a lot of individual variation between organisms. Okay, and so now moving on to the results for the onset of symbiosis, and so we also call this kind of colonization. So for this, so these are the results of APDF1, APDF2B, and APMAPs with time post-infection and relative quantities. And so you can see here that the only significant difference is that APDF2B is upregulated at 24 hours post-infection in comparison to the no symbiont control. So the symbiont is the solid line, and the no symbiont controls are these dotted lines. Another interesting kind of observation from this is that there's this decrease or dip in expression for all of the no symbiont controls for all 3 genes at the 24-hour post-challenge time point. And so you can see that in all 3 of these graphs. And the reason for this is unknown, but it really seems like there's something, this comes out across multiple experiments, something happening at that 24-hour time point. And so overall, kind of what we concluded from this is that APDF2B seems to be upregulated at the onset of symbiosis, but because the differences weren't significant for APDF1 or APMAP, their role in symbiosis remains unclear. And so this kind of provides yet another piece of evidence that APDF2B is involved in symbiosis. And so now I'd like to kind of conclude chapter 2 by looking at tissue layers and tissue-specific expression. And so for this objective, I use a technique called laser capture microdissection. And so this is a technique in which you can cut kind of defined regions of a tissue section out so that you can isolate specific tissues or cells, and then catapult them into a collection tube. So you cut them out with a laser that's kind of associated with the microscope. And I just want to kind of highlight Nate Kirk in this was a big part in kind of generating ideas for this project and also helping out the microscopy aspect. lead us to think that this would be a good marker for the epidermis. And then for the gastrodermis, I used a Symbiodinium-specific primer set. And so the results for this are right here. And so you can see that although there is some signal in the epidermis, it's much stronger in the gastrodermis. And so these were the marker genes that I used in this experiment. So with those in hand, I then performed qPCR on both these tissue marker genes to get kind of a more precise measurement of their expression, and also APDF1, APDF2B, and AP mouse. And so what did I find in this? Well, the first thing that was very apparent is that you get really, really small amounts of RNA with this technique. And so this is a real limitation. You spend a lot of time collecting this tissue, and then you get very small amounts. And so this really limits kind of the downstream application that you can perform. And so because of this, only 2 of my samples, when I carried it through cDNA synthesis, were actually usable, as in they produced usable CT values, so the other ones were just too low in concentration. And so the graphs I'm about to show you are based on a sample size of 2, and so obviously I'll have to go back and collect more samples for these. And so this top graph here represents results for the tissue marker. And you can see that they're really just not quite as pretty as they were before. So NG is hematoelective again, the epidermis marker. And so you can see that there's really no difference in expression between the tissue layers. And so this is kind of a bummer. But we think this may be due in part because nematocysts can be present in the gastrodermal tissue. And so it could just be dependent on kind of the area within the animal that you're sectioning. And so more work, if someone wants to pursue this technique further, will need to be done to kind of confirm a better epidermis marker. The Symbiodinium marker here shows the general pattern that we would expect. So you have higher expression in the gastrodermis and the epidermis. And so I still felt confident in the tissue that we had isolated, whether it was epidermis or gastrodermis. And so then for the actual genes of interest, the complement genes, so you'll notice that I only have results for APDF1 and APDF2B here. So MAS in general shows kind of lower expression levels than the factor B genes. And so again, because these were really low concentration samples, they were pretty much undetectable in these samples. So I don't have any data to show you on that. But what you can see is that although APDF1 doesn't really show any patterns, APDF2B, there's kind of evidence to suggest here that it is more highly expressed in the gastrodermis than in the epidermis. So this again is exciting, and it kind of gives you a level of information you can't get from a simple symbiotic state comparison, and serves as yet kind of another piece of evidence that APDF2B is involved in symbiosis. But again, this is kind of just suggested at this point, and I'm planning this fall to go back and do some more of this in hopes of getting some more data points. So just to summarize kind of all the work in the symbiosis chapter, and so for objective 1, we saw that Atesia has 3 factor B and 1 MAS transcript. In objective 2, phylogenetic analysis was performed, suggesting that there were kind of these different groups of Nigerian sequences for factor B on the tree. And I wonder whether this was due to some sort of functional divergence. And then the functional work, objective 3. So bringing back, we're going to kind of be using this model throughout the talk. So we showed that for interactions with Symbiodinium, APDF2B seems to be pretty important. And so we have 3 lines of evidence to suggest that it may be playing some role in symbiosis, in that it was upregulated at the onset of symbiosis, it's decreased in the symbiotic state, but when you look at the layer of the tissue levels, you can see that it's more highly expressed in the gastrodermis where the symbionts are residing. And so all of these 3 pieces of large changes in factor B or mass expression. And so these are the results for LPS. And so you can see that all three genes show this kind of general trend of downregulation at 12 hours and then increase for the duration of the experiment. However, there were no significant differences between any of the time points and time zero, or either there was a DMSO or vehicle control incorporated at 24 hours, and there was no significant differences here as well. But there were significant differences between for APBF1 and APMAS between the 72-hour and 12-hour time point. But kind of the conclusions that you can draw from that are limited. For peptidoglycan, we saw kind of a similar story. And so here is our 0-hour time point, and then 3 kind of treatments at 24. So the DMSO control, the low concentration, and the high concentration. So here as well, the treatments were not significantly different from either 0 or the DMSO control. But you see this kind of general trend of greater expression with the low concentration treatment, so the 10 micrograms per ml, and then decreased concentration for the 50 micrograms per ml. And there was a significant difference for APMAS between this 10 and 50 microgram per ml treatment. But again, kind of the conclusions that can be drawn from this are limited. And so right after we had finished these experiments, a really cool paper in Horseshoe Crab came out that kind of demonstrated a similar result in which they challenged with a wide variety of individual immune stimulants and looked at factor B protein activation. Didn't really see much, but then when they challenged gram-negative, gram-positive bacteria, they produced a pretty robust complement response. And so this kind of confirmed the result that I saw. And so because of what they saw, I also wanted to try this challenge with live bacteria. And so again, I used ratiomarcescens as my infective agent. And so for this experiment, I used both ecosymbiotic and symbiotic animals. And so I wanted to see whether symbiotic state had kind of any bearing on the response that we saw. And so I started, these animals were kept in kind of their natural environment in the lab. And so ecosymbiotic animals are generally kept in the dark. And so I maintained them in the dark for the experiment, and then the symbiotic in the light. And so I activated them on shaker tables for about 24 hours because they were kept on these for the duration of the experiment, and then treated with 3 different concentrations based on kind of that preliminary work that had been shown that 10 to the 8th really caused the aphasia to look sick in a sense, and so I didn't want to come anywhere close to that. I was just hoping to generate an immune response. And so I sampled these 3 treatments at 0, 24, and 48 hours post challenge, and then performed qTCR on factor B and MAS genes. And so I got a lot of really cool results from this experiment. So in contrast to the immune stimulants, I actually saw dramatic changes in expression here. And so I'm going to walk you through this kind of gene by gene. And so these are the results for APBF1, and we'll see that the response depends on both bacterial concentration and symbiotic state. And so this is kind of true for all the genes that we'll look at. And so for symbiotic animals here, we can see that there's a lot of action going on here at the 24-hour time point, and that there's significant upregulation in both the low and the high concentration treatments. For the aposymbiotic animals, we only see upregulation for the low concentration treatment. And so we begin to see this trend that symbiotic animals tend to be more responsive with a low bacterial concentration treatment. APBF2B at that same time point was downregulated. And so this was all really exciting to me because it really kind of gets at this functional divergence in which APBF2B may be more kind of symbiosis related, and APBF1 and APMAS may be working against pathogenic microbes. And so if we kind of go back to the beginning and think about the tree that I presented to you before, this also kind of matches up with these results. And so APBF1 is part of this BF1 group that I showed you before that groups with those deuterostome sequences. And though there has been some functional work performed on them, and they do respond to pathogenic microbes. And so this kind of matches the result that we got for APBF1 and APMAS. And then in contrast, APBF2B is part of this kind of Nigerian-specific group over here with the APBF2 sequences. And we saw that it was kind of symbiosis-specific, and so this may be something novel that Nigerians have evolved to interact with both the negative and positive associations that they encounter. And so with that, I have a lot of acknowledgments. First and foremost, I'd like to acknowledge my advisor, Virginia White. She has been extremely supportive in both my research and my teaching endeavors over the last 6 years, and really encouraging me in that kind of perseverance aspect. So it's definitely been a long road. I'd also like to thank all of my committee members for being very supportive over the years, and also taking a vested interest in my research, even though oftentimes their research is pretty far removed from our study system. And so thank you to Mike Lewin, Michael Freitag on Skype here, Eli Meyer, Kathy Magnuson, and also Luis Bermudez spent some time serving on my committee and was very generous in both his time and lab space. The IB department has been a great department to be a part of. Our office staff are awesome. The 4 Ts have always been there with any questions that I have and for support. Also thanks to Joe Beatty and Bob Mason for arranging all of the TA shifts. And I also wanted to give special thanks to Devin Quick for inspiring both my interest in teaching and also in human anatomy and physiology. Funding sources come from the National Science Foundation and the generous resort funds for the IB department. And then I also want to give a special thanks to all of the Vice Lab members, past and present, and all of the undergraduates who have really made my research possible. Specifically, Ellen Dow did a lot of really good research while she was here in the lab, and I really appreciate her dedication and also her perseverance in the lab. And so, of course, I have lots of pictures to accompany this. And so being in the White Lab has been a really fun experience. It's been a really supportive research environment, and we've also had a lot of fun along the way. I mean, most labs don't have a pet sea cucumber. For me, I was passed away a couple years ago, but just wanted to bring it back here. And so it's been a really positive experience to work in the lab. And I want to give special thanks to Sheila Kitchen. She's been my partner in crime, both inside and outside of the lab, while she's been here. And when she was interviewing, actually, I told Virginia, you know, you may be interviewing for your new grad student, but I'm interviewing for my new best friend. And so this turned out to be very true, turned out perfectly. And so we've had a lot of fun inside the lab, collaborating on projects. We wrote a lab email together, so that was a big undertaking. We pretend to be runners. And we dress up as our study system for Halloween. So it doesn't get much better than that. And then I developed a really supportive community in Corvallis over the last 6 years. It took a while, but I made some really awesome friends, both within the department, outside of the department,
talk that you said that arthropods didn't have complement.
So some arthropods do.
Okay,
So
because then you
some
showed
are
your...
insects. I haven't found any complement sequences in insects or crustaceans, but members of Chelicerata. So there's been several studies on horseshoe complement, and also ticks, mites, spiders all seem to have.
Okay, that's where I was confused, because then you showed the horseshoe crab one.
Yeah.
Okay, well my question is, during your bacterial challenge experiments, did you see a difference in mortality between the APOs and the non-APOs?
So I didn't observe any mortality, and I specifically chose concentrations that I didn't think would induce mortality. And so my animals actually looked pretty healthy during the entire duration of the experiment. And so previous work that had been done in Serratia showed that basically when you got to a concentration of about 10 to the 80 cells per mil, that's when things really started going downhill. But before that, from just kind of a macroscopic level, they look pretty healthy.
I think that's interesting because you might imagine that the presence of the conspicuous symbiont might essentially create a situation which they can't respond to bacterial challenges and might diminish the utility of the symbiosis in terms of bacterial pathogenesis.
Yeah, and I mean, Serratia has been shown to induce loss of symbionts and kind of disruption of the gastrodermal tissue. And so I think maybe a little bit what we saw at the end of that upregulation of APDF2B is maybe interactions between Serratia and also this bleaching response, even though I didn't really see bleaching.
I was curious about Symbiodinium and whether or not they're considered more of a passive member of the symbiosis, especially during the onset and continuation of the interaction, or if there's some kind of something going on with them that facilitates the symbiosis.
Do you need, I mean, they're definitely a vital part of the kind of microbial community of coral.
Right, but on a molecular level, are they doing anything to facilitate the onset of the symbiosis?
Oh,
Or are they just being taken on by the coral and the coral is
yeah,
the one
so some
dominating?
of the innate immune pathways that I kind of flashed up at the beginning, so there has been several lines of evidence to suggest that there's interactions between host lectins, so these would be host kind of receptors, and then glycans or sugars that are present on the symbiotic surface as being really important kind of interactions. And so there definitely are interactions between innate immune system of the host and then molecules that are present on Symbiodinium that are really important at that onset of symbiosis stage.
Okay, but Symbiodinium's not like producing some chemical.
I mean, they definitely could be.
Okay.
Great. Kind of follow up on some of those questions. So when you were doing the FPS, effective right-brain exposure, those were asymbiotic open.
Yes.
So do you think it might, maybe the symbiont is contributing to the immune response of the host or of the...
Yeah. Absolutely. And so the reason that I chose the asymbiotic animals was previous work had been done by a postdoc, Olivier Defournet, in the lab
slides to the trigger real quickly? Because I thought this is really kind of interesting. Yes. So I thought this was a cool idea that maybe this gene family originally used for sort of pathogen defense gets co-opted to help facilitate this important symbiosis. So if that's true, then if you mapped cnidarians or just invertebrates more broadly, they can establish symbiosis with diamond flagellates on the tree and reconstruct when those symbioses were supposed to have occurred. Could you kind of match that up to the presence of your gene family? And then the sort of follow-on to that is in sort of aposymbiotic or azoxanthellate lineages where they just don't normally associate with somebody. It would be interesting to see whether maybe you don't actually lose a copy of that gene, but maybe the sequence diversifies a little bit more, like there's a release from selection or something.
An interesting observation that I made is that most cnidarians tend to have 3 factor B transcripts, whereas Nematocella, which is a non-symbiotic organism, only has 2. And so obviously this is just one organism and one line of evidence, but maybe there is something to that. And yeah, I mean, it would definitely be interesting to tweak all of, I guess the problem is that there's really been no other work on complement that's done in kind of a beneficial association capacity. They also get some challenging with immune stimulants or immune challenge. And so I would say that I don't really have the information currently to kind of reconstruct that on a tree, but it's a really interesting hypothesis to investigate.
One more question for Chris.
This really goes back to the broader picture, if you don't mind. I was interested in the evolution of the complement system, of course, and in one of the tables early on in your talk, you showed about 3 major components of the system, including the mast at the end there, which is, of course, mannan-binding lectin-associated serine proteins. The fact that mast works by being activated by mannan-binding lectin, which has to bind to mannans on targets, did you look to see whether those species or those taxonomic groups that have mast also produce MBL or a homologue of MBL?
I never specifically looked at it in that capacity. I mean, there are there are definitely mannose binding lectins present in cnidarians, and so, but whether they're actually associating with the masts, I don't know.
Is that functional evidence of mannan-binding lectins being present, or are they being found in the genome?
No, so there is mannose binding